about the mitosis cell division in brief. Today we will solve a question based on the mitotic cell division. Okay, that of course I think the two questions we discussed today. Let us start from the beginning, and this is say here that our first question number one is cell which are not dividing are likely to be at which stage? They are at the G1, G2, G0 and S phase. In which phase? Please tell quickly that which
G
one is
naught,
correct. Yes,
G naught phase.
3 G naught. Okay, G naught, G naught, G0 or G naught stays. Here it is those cells which are not dividing. They undergo the process of maturation, and they come out of the cell cycle. Here you can see that this is the cell cycle. This after the division of the completion of the mitosis, cell are two cells are formed. These two daughter cells, if it is not dividing hmm, any further, then they will come out from the G1 and from the cell cycle, and they are in the G0 stage. G0 stage, these are actually the mature cell. Okay, mature, and they are differentiated. They are specialized. Okay, specialized for specialized for a specific function. Is it a specific function? So in the if we see in the plant cell, okay, those permanent tissue, no, this all the permanent tissue, they are permanent tissue. They belongs to this G0 phase. Okay. Sometime when this is again, there is a need that they should divide, then again this from this G0 phase, they will come back and join the S phase. They will join S phase and they will replicate the DNA and enter into the cell cycle. So this is now it is the number two question. Number two, they select the correct statement about G1 phase. G1 phase, what is this? That the correct statement about cell is metabolically not active. Okay, DNA in the cell does not replicate. It is not a phase of synthesis of here. Actually something this has been deleted. Synthesis of mm, DNA. Okay, this one you read, it is not a phase of synthesis of, it is not a phase of synthesis of, no, not a DNA. This DNA does not replicate. It is phase of synthesis of the cell organelles. Cell organelles Students,
Yes,
hello, you can hear me, no? There
sir.
was no current, so that's why some disturbance in the. Maybe it will be a problem for the laptop also. Okay, the question number two, that it is they correct about the G1. G1 is cell is not metabolically active. This DNA in this cell does not replicate. It is not a phase of synthesis of cell organelles or cell macromolecules. Okay. Cell stop growing. Which one is correct for the G1? Can you say which is correct for the G1? Think quickly.
It is not the field of synthesis of cell organelle.
Yes, it is. Say this one, I cannot hear you properly. This one, that the option 2, the DNA in the cell does not replicate. In the G1, the cell is metabolically active because during the when the two cells are formed, this young cell grow. Young cell will grow, and growing is, that is the synthesis of protoplasm, is it? Synthesis of protoplasm, all the proteins, RNA, these things are synthesized, new cell organelles are produced. So this cell is at G1 phase, the cell is very active metabolically. Only thing that it will not replicate the DNA, because still then it is not decided whether this will go for the further division. First, it has to grow. So, growing takes place during this phase. All other organelles also are synthesized, macromolecules synthesized. Cell stop growing. No, cell continue grow in terms of protoplasm synthesis. So, our answer is the cell in the DNA in the cells does not grow in the G1 phase. Next question, I think this one we have discussed that in the previous class, replication of DNA is a prerequisite for eukaryotic cell to undergo division. During the cell cycle, DNA replicate at S phase, is it? DNA replicate at S phase. So this is not the this is previous days lines, etc. There, this was asked in the AFMC 2007. Next question, that next question also, I think we have discussed the plant cell can show mitotic division in plant cell can show mitotic division in the diploid as well as haploid cell. Diploid cell can divide, haploid cell can divide, and also polyploid cells can divide by mitosis. So our answer will be both, that is diploid and haploid cell can. In animals, in actually in the higher animals, living only very few, that lower group of animal, all the animals are usually, they are diploid organism. All animals are diploid organism, and in them the mitosis, meiosis, both these takes place in the diploid cell. But in case of plant, diploid, haploid, all the cell divide by this polyploids also. So answer is this one. Next question. This is 3, 4. Okay. ঋষভ, আবার এই যে কারেন্ট আসলে আবার কি আলাদা করে আবার জয়েন করেন? নেক্সট কোশ্চেন উইল question number 5. A somatic cell that has just completed S phase, okay, S phase has been completed of its cell cycle as compared to gamete of the same species. The cell has completed S phase, okay, and now if we compare this cell with the gamete, okay, gamete of the same species, then what is true for this? The twice the number of chromosome and twice the amount of DNA of the same species. It has twice the number of chromosome and twice the amount of DNA, or same number of chromosome but twice the amount of DNA, twice the number of chromosome and 4 times the amount of DNA, or it is 4 times the number of chromosome and twice the amount of DNA. Here, which one will be correct? See, we will take it like this, that the cell is a diploid. Is it? In the diploid cell, the chromosome number is 2n, chromatin is 2c. Is it? 2c is the chromosome. And if we take the gamete of that, then in the gamete cells are haploid. It has one number of chromosome, one set of chromosome, and 1c amount of DNA. Here it is 2c amount of DNA. So in normal case, what happens that the number of chromosome in compared to the gamete is double in a diploid cell. Amount of DNA is double in compared to the gamete. All right. Now this cell has already completed S phase. Already completed S phase. This one already completed S phase means the DNA has been doubled. That is, DNA replication has taken place, and so the DNA has become 4C. But what happened? The number of chromosome does not change. So the number of chromosome remain double in compared to the gamete. Chromosome number is double, 2 times, but the DNA amount is 4 times of the gamete. So it is the same number of chromosome, no, twice the number of chromosome, okay, and 4 times the amount of DNA. The cell which has already completed S phase, they will have double number of chromosome, that is the diploid number, and 4 times the amount of DNA will be there. So our answer is this option number 3. Now come to number Sixth question. During which phase of cell cycle amount of DNA in a cell remain 4C level if the initial amount is 2C? Say initial amount is 2C, 4C it become when? During the S phase it become double. So in the S phase it will become 4C. After S phase comes G2, that also will have 4C. In the prophase of mitosis it will remain as 4C. In the metaphase it will remain as 4C. In anaphase the cell will have 4C amount of chromosome, but at telophase when this cytokinesis also will take place, then again the amount of DNA will reduce to 2C. So these are the cases that where it will have 4C level of DNA. So it is written G0 and G1. No, 4C level of DNA is not in the G0 and G1, because in the G1 DNA synthesis does not take place. G0, the cell is not within the cell cycle, it will not divide. So there is no DNA synthesis. So it will remain as 2C only. Here it will be 2C. G1 and S phase. See here G1 also written and S also written. In the S phase it will become 4C, but at G1 it will not. So this answer also we cannot say. This is not correct. Only G2, and here it is given G2 and M. Now in this 4C level of DNA we will find in G2, and also in the M, not in all prophase metaphase. Certainly we will get prophase metaphase anaphase, we will get 4C level, but at telophase, which is also part of the M, here it is not correct. So we cannot tick this option 4. We have only one option left, that is 3. Only G2 from all these options. Okay. Otherwise, this double amount of DNA content will be in the S phase, G2, after the S phase, completion of the S phase, G2, prophase, metaphase, anaphase. This is CBSE AIPMT 2014 question. Question number 7. So which of the following is the best stage to observe the shape, size, and number of chromosome in a cell? It is the metaphase. In the metaphase, chromosomes are distinct. The condensation of the chromosome has taken place, enough sufficient condensation, and at this stage, all the chromosomes are not scattered. They have aligned along the equatorial plane. So counting of this chromosome is also possible, and their shape, size, what type of shape is there, that we can study here. So it is the metaphase of the metaphase of mitosis. Okay, not the metaphase of meiosis. For the chromosome morphology, metaphase of mitosis is studied. Next one, a certain species of animal has 6 pairs of chromosome. Animal has 6 pairs of chromosome. 6 pair means 2n is equal to 12. Okay. How many molecule of DNA do the nuclei of this animal have during G2 phase? During G2 phase, how many molecule of DNA? You should remember that the cell which is not dividing, one chromosome, one chromosome is equal to one molecule of DNA. Because the chromosome is, each chromosome is having one chromatid. Means one chromosome is equal to one chromatid, and it is made of one DNA molecule. Okay. So if, but it is, the cell is in the G2 phase. Now what happened in the G2 phase means already DNA synthesis, the DNA has duplicated. The molecule of DNA has been duplicated. So from one DNA, two DNA molecule has been formed. Right. So if the cell is having initially, when it is not dividing, it has 12 chromosome, means 12 chromatid, means 12 DNA, 12 DNA. But as S phase has completed, so in the G2 phase, it will have double the number of DNA. So it will have 24 DNA molecule. Okay? It has 24 DNA molecule. Clear? Say this in the prophase. In prophase, each chromosome is with two chromatids. Means each chromosome is having two DNA molecule. In metaphase also the same, but in anaphase this chromatid separates, sister chromatids. And now in the anaphase to telophase, there one chromosome means one DNA molecule. One chromosome, one chromatid, one DNA molecule. Next one, question number 9. Mitotic spindle fiber. During mitosis, there is thin thread, very thin threads developed between the pole, between the poles, and this thread is known as spindle fiber. Is it? These spindle fibers are made of protein, and this is made of protein. That protein is known as tubulin. Okay, and they form, this protein forms as the tubes. These tubes are they are microscopic, very thin, so they are known as microtubules. Okay, microtubules. So the spindle fibers composed of microtubules. not centromere, not centrosome, not kinetochores. Number 10, which in which of the following mitosis is not used? In which of the following mitosis does not take place? Where mitosis is not taking place? Say repair of wounds in multicellular organism. This healing of wound means new cells are formed. This thing is carried out by mitosis cell division. So here mitosis is used. Asexual reproduction in unicellular organism. Asexual reproduction of unicellular organisms also takes place by mitosis. Development, that is it, baby in mother's womb. Our life start from the single cell. Single cell, that cell is called zygote, is it? So from the this single cell multicellular organism we are growing, this is due to mitosis. So growth, development is also by mitosis. Okay. Production of gamete. This production of gamete is not by mitosis. This production of gamete takes place by the division called meiosis. Is it? So this one here during the production of gamete, the mitosis division is not used. It is by the meiosis because during the gametes are haploid and they here the chromosome number is reduced to half, so it is by the mitosis. During which stage of mitosis do centromere split? In animal cell, oh sorry, it is centromere. Centromeres are a structure of what? Centromere. Centromere is the site where the two chromatids of a chromosomes are attached. This is centromere, is it? And at the centromere there is kinetochore to which the spindle fibers are attached. So this centromere during anaphase, during anaphase, this centromere will split longitudinally and the two chromatids will separate. Okay, so two chromatid, one and these two chromatid will separate by longitudinal division of the centromere. This is in normal case centromere divide longitudinally. So two chromatids are separated. Now these chromatids, they are known as daughter chromosome. Daughter, now they are daughter chromosome. Okay. So daughter chromosome separated due to the longitudinal division of the centromere. Sometime due to the abnormality, this centromere may divide transversely. If they divide transversely, then a abnormal condition is produced. Both the chromatids, these two halves, which are identical, they will go to the same side. Next question, during which stage of mitosis does cytokinesis usually occur in animal? Which stage of mitosis? During which stage of mitosis cytokinesis occur? This is telophase. Okay. But the process of cytokinesis start towards the late anaphase. But in the telophase, it is completed. Next question. At which stage of cell cycle are histone proteins synthesized in eukaryotic cell? See, histone proteins are special proteins. This one is associated with the DNA. DNA plus histone. This give rise to the structure which is called chromatin. This complex is called chromatin, and this chromatin make the chromosome chromatids. All right, chromatin thread. So along with the DNA synthesis during the S phase only, most of the histones are synthesized, but at the later phase also it is also synthesized. So DNA histone synthesis takes place in the during the S phase only. All right. Sorry, this one is I think this is not the 2050. This is a just a mistake here. 2005 it is, right? Next question. Number 14. Centromere is required for transcription, crossing over, cytoplasmic cleavage, movement of chromosome towards the pole. There's also CBSC. See here also this mistake has taken place. Hmm, this is a mistake. This is, please read it as 2000. Okay, uh, this one is this is 2005. Centromere is required for what? Transcription. Do you know what is transcription? Transcription is, this one is called, that is synthesis of RNA. This is known as transcription. And crossing over, that is exchange of segment. This one is exchange of segment of chromatid. It's between non-sister chromosome. Non-sister chromosome. All right. That homologous between, or non-sister, you can write between homologous chromosome. Okay. Among the non-sister chromatids, we can say. So here crossing over is this cytoplasmic cleavage. Cytoplasmic cleavage means it is just like cytokinesis. The centromere is required for the movement of chromosome towards the pole. If a chromosome without centromere mere does not survive, okay, this is chromosome without centromere. It is called acentric chromosome also. This is acentric chromosome. This one does not survive. Centromere is a must. At the centromere, the spindle fibers are attached. Is it? At the centromere, this spindle fibers are attached, and on the centromere, there is kinetochore. Kinetochore to this spindle fibers are attached. So this the centromere is, presence of centromere is essential. Otherwise, chromosome will not move the opposite pole after the this anaphase. So this is required for the movement of chromosome towards opposite pole. This is also CBSE IPMT 2005. Question number 15. Which statement about daughter cell of mitosis is correct? In mitosis, from one mother cell, after division, two daughter cells are produced, and these daughter cells are identical to each other and to the mother cell, and they have same amount of chromosome and the same amount of DNA, type of DNA, so they are identical. So what is correct? One of the two daughter cell is genetically identical to the parent cell. No, one of the, here it has been told one. No, both actually. So this is not correct. They differ genetically from one another and the parent cell. It is not correct. Identical, similar. They are genetically identical to one another and to the parent cell. So this one is correct for the daughter cell produced in the mitotic division. They are genetically identical to one another but different from parent cell. No. Okay. So number 3, this one is correct. Now question number 16. What are the most important structure involved in moving chromosome during mitosis? What is important? Most important moving chromosome during the mitosis. Here kinetochores and chromosome kinetochores and mitotic spindle. This one is required because mitotic spindle attached at the kinetochore of centromere. Okay, so spindle fiber is required. Spindle fiber pull it towards the pole, and spindle fiber is attached to the kinetochore of the centromere. Kinetochore is present on the centromere. So this is 16. This is 17. Okay, in between, I think you can see one question that is based on the diagram. See this diagram. Can you say clearly the diagram? Here, identify the phase of mitotic cell division from the given diagram. This one is which stage? Prophase, metaphase, anaphase, telophase. Which stage it is?
Anaphase.
Anaphase. Here it is the anaphase. So this one will be the answer, because here we can see that spindle fibers are there and the chromosomes, this they have separated each chromosome with one chromatid, and it is moving towards the opposite pole. So it is anaphase. Okay, come to question number 17. In the somatic cell cycle, G1 phase DNA content is double the amount of DNA present in the original cell. Is it correct? No, this is wrong, because in the G1 phase, DNA synthesis does not take place. DNA replication takes place in S phase. This is a correct option. Okay. DNA replication takes place in the S phase. Hmm. A short interphase is followed by a long mitotic phase. A short interphase is followed by a long mitotic phase. Mitotic phase is never longer than the interphase. Interphase is usually longer. So this is also not correct. G2 phase follows mitotic phase. No. G2 phase, mitotic phase follows G2. Okay. This is CBSE IPMT 2004. 18. Synthesis of RNA and protein takes place. Synthesis of RNA and protein takes place in which phase of cell cycle? Synthesis of RNA, not S phase, M phase, no, G1 and G2 phase. This is G1 phase, that is before the DNA synthesis, when the young cell is growing. So in the G1 phase and G2 phase also, there is DNA and RNA synthesis. Sorry, protein and RNA synthesis takes place. The statement for mitosis is cell formed by it perform diverse function, that is show division of labor. Two cells formed as a result of this division are identical in all respect. Yes, this is also true. Cell formed by it have half number of chromosome than that of parent cell. This is not true for the mitosis. Cell formed as a result of mitosis have different genetic character. This is also not true. Okay. By it perform diverse function. Here only a statement for mitosis is the Here I think question is a little bit incomplete, I think typing mistake. The correct statement, a statement for mitosis is that the two cells formed as a result of this division are identical in all respect. Yes, this is true. Okay. Division of labor, that also not necessary that they will diverse function, they may not do, they maybe have the same function. 20. In animal cell during the S phase, the DNA replication begin in the dash and centriole duplicate in the dash. So where it is the DNA replication in the nucleus, okay, and at the in the S phase in animal cell the centriole also replicate, and centriole is present outside the nucleus, so it is in the cytoplasm. So first one here it will be nucleus and here it will be cytoplasm. So it is the nucleus and cytoplasm. Following event represent which stage of mitosis? Say here two statement has been given, A and B. Two statements. Centromere is split and chromatid separate. This takes place when? Splitting of centromere and movement of chromatids of the chromosome separate. Chromatids move to opposite pole. See, these are the two key character or anaphase of mitosis. Is it? This is anaphase. This in the anaphase, if you write only these two characters, then also it is very character, two main features. 22. The plane of alignment of chromosome at metaphase is referred as attachment plate, cell plate, metaphasic plate. Is it? When the chromosomes so they are aligned, okay, at the equator, equatorial plane, and this structure we call that metaphasic plate, right? Next, mitosis is usually restricted to the dash cell only, degenerate, diploid, haploid, polyploid. See here, mitosis is usually restricted in diploid cells. You have to go. But in particularly in animal and in plant it is both. But mostly our higher organisms, all our higher organism, whether it is plant and animal, they are diploid. Is it? They are diploid. So maximum this cell division, they are taking place in the diploid cells. In animals, it is all the organisms are diploid, excepting a few lower group of animal. Number 24, which of the following is not a significance of mitosis? Mitosis has several significance, which one is not? Here it increases the genetic variability in the population of organism from one generation to the next. It results in the production of diploid daughter cells with identical genetical complements. Usually, the growth of multicellular organism is due to mitosis. This is true, and cell repair is due to the mitosis. This is the result in the identical genetic complement cells, daughter cells with identical. So all these three, option number 2, 3, 4 are the function of function or significance of mitosis. But this one, option 1, okay, this is not the significance of mitosis, but it is significance of meiosis. 25. Which of the following is not the characteristics of mitosis in somatic cell? Again, similar question. Disappearance of nucleolus, chromosome movement, synapses, spindle fiber. So the spindle fiber is takes place formation, the chromosome movements also takes place, nucleolus also disappear in the mitosis or in the somatic cell. But in the somatic cell, or that is the body cell, there is synapses does not take place. Synapses is a process, very, very important process in the meiosis. And meiosis takes place, that is in the germ cell during the formation of gamete. Germ cell or the germ line cells which ultimately give rise to the formation of the gamete. Of course, in case of plant, there is initially there is production of spores. During the production of spores, spores are haploid. During the production of spores, that is microspore, pollen grain, this meiosis takes place. This is the NEET 2016 question. 26. The complete disintegration of nuclear envelope in a cell cycle marks the start of prophase, start of metaphase, end of anaphase, start of telophase. This is the beginning of a start of metaphase. This one, the complete disintegration of nuclear envelope is the start of metaphase, or this is end of prophase. Okay, where prophase end, metaphase start. This cell division, these things are not abstract event. This is a continuous process. Question number 27. At which stage of mitosis the two daughter chromatids separate from each other, migrate towards the opposite poles, and are now referred as the chromosome of the fused daughter nuclei? At which stage? It is the anaphase. No? Chromatids separate, and then 28. Choose the correct sequence of two main events in mitosis. Correct sequence, that is karyokinesis followed by cytokinesis. It is two main events are here: cytokinesis followed by karyokinesis, karyokinesis followed by separation of daughter cells, cytokinesis followed by separation. Two main events are, one is main event of mitosis. Mitosis we can study in two main phases. One is karyokinesis, and then it is cytokinesis. is it? Here the nuclear division and here it is cytoplasmic division. So cytokinesis followed by no, not cytokinesis followed by karyokinesis. Cytokinesis takes place later on. Say here in this option 1 and 2, you have to be very careful which one is coming first, which one is coming later on. Both cytokinesis and karyokinesis is there, but first the karyokinesis takes place and then cytokinesis. So our option 1 will be the correct sequence. This is karyokinesis followed by cytokinesis. This is 29. If one question we see this one also based on the diagram, similar type. Okay, here given diagram indicate which of the following phase of mitosis. Choose the correct option. So here it is. The correct option is this one is anaphase. It is movement of the chromosome to the opposite pole, and they are reaching the other one question during anaphasic movement of chromosome. Centromere of each chromosome is towards centromere. Centromere of the chromosome is which side? See, this is the chromatid. Centromere is here because the spindle fiber is pulling. So centromere is towards the pole. It is towards the pole. So this is 32. This one will be 31. Hmm. Which of the following statements are correct for the cell cycle? Cell cycle is the sequence of event involving, this is the definition of the cell cycle. Cell growth. Cell growth in terms of is a continuous process. This is also correct. This is also DNA synthesis occur only during one specific stage in the cell cycle. Yes, this is also correct. The replicated chromosome, that is the DNA, are distributed to the daughter nuclei during the cell division. This is also true. So our this option codes are here 1, 2, 3, 4, all are correct. Okay. Read the following statement and select the correct option. M phase represent or twice written as represent. The phase when the actual cell division or mitosis occur. Yes, this true cell cycle in yeast takes place about that 90 minutes it takes. Yes, this is also true. In the 24-hour of average duration of cell cycle, human cell division proper lasts for only about 1 hour. This is also true. M phase lasts more than 95% of the duration of cell cycle. No, this one is wrong. M phase take very less. This is the interface take 95% of the time. Okay, so that 1, 2, 3 is correct, but the number 4 is not correct. So here one question, I think it is already time. So these are the question for the matching. Here matching type, match the column. Column 1, separation of daughter chromosome takes place when? Separation of the daughter chromosome, interphase, karyokinesis, S phase, cytokinesis, where we should match this with 2, karyokinesis. Separation of daughter chromosome, division of cytoplasm, That one we call them cytokinesis, is it? Phase between the two successive M phase. This is the interphase, and synthesis phase is S phase. So this way we have to match also. Okay, today we will stop here.